In the second part of this lecture, I would like to talk about catalytic strategies such as acid-base and covalent catalysis. And the first in the list is general acid-base catalysis. You must be familiar with this type of catalysis from organic chemistry, and one example of acid-base catalysis is shown on this slide. It's hydrolysis of amide bond. So acid catalysis involves protonation of the substrate, and specifically in this case we have protonation of oxygen of the amide bond, which increases electrophilicity of carbon, so with a full charge on carbon. Now oxygen of water molecule can easily attack this carbon, leading eventually to hydrolysis of amide bond to get carboxylic acid and amine. There are several amino acid side chains that participate in acid-base catalysis, and they are listed here on the slide: aspartic acid, glutamic acid, histidine, cysteine, tyrosine, lysine. For example, aspartic acid and glutamic acid are deprotonated under physiological conditions, and they can act as general base catalysts, meaning that they can remove proton from a substrate and increase negative charge on the substrate by thus increasing its nucleophilicity. A good example of general acid and base catalysis used by the enzyme is ribonuclease A. This enzyme cleaves single-stranded RNA by hydrolyzing its phosphoester bond, and it's quite efficient. It increases reaction rate by 12 orders of magnitude. RNase A has two catalytically essential amino acids, histidine 12 and histidine 119. And what you see here is the substrate of RNase A bound in it between the two histidines, and histidine 12 is deprotonated, and it can act as a general base to pull proton from 2' OH group of the first nucleotide, which increases nucleophilicity of this 2' OH group and makes it attack the phosphorus. On the other hand, histidine 119 is protonated. You can see positive charge on it, and it can donate proton to 5' oxygen of second nucleotide and make this nucleotide a good leaving group. So after the cleavage of the bond is completed, the 5' portion of the RNA is released from the active site, and water enters the active site. And at the second stage of phosphodiester bond hydrolysis, histidine 12 is deprotonated and can pull proton from water molecule and thus increase its nucleophilicity towards phosphorus. And histidine 12 acts as an acid. It protonates 2' OH group and makes it a good leaving group. So on the end you have 3' phosphate group and 2' OH group as a product, which is not shown here. So you can see that histidine can show amphoteric properties, which means that it can act in active sites of enzyme both as an acid as a base. This slide shows you three-dimensional X-ray crystallographic data of ribonuclease A in complex with its substrate. so this is dinucleotide substrate, you can identify the two catalytically essential histidine residues that are positioned next to the cleavage bond. And what is interesting that not only the two histidines participate in catalytic action, but also other amino acids such as this lysine residue that binds phosphate group and OH group of the nucleotide by hydrogen bonds also is essential in positioning the substrate next to the catalytically essential amino acids. Next in the list is covalent or nucleophilic catalysis, which accelerates reaction rates through the transient formation of catalyst substrate covalent bond. The amino acid side chains that are capable in participating in covalent catalysis are shown on this slide. These are OH groups, first of all, and they can produce. They can be deprotonated to produce O minus, and this alkyl halide ion is very nucleophilic. Thiol groups, amino groups, and imidazole group in histidine. In fact, you can assess the strength of a nucleophilic group by comparing the pKa of the acid base, of the conjugated acid and base. The pKa, for example, for serine, for OH group and serine and threonine are above 14, so which makes them the strongest nucleophiles among all groups found in amino acid side chains. Histidine is the weakest nucleophile, even though it's also has nucleophilic properties. I would like to demonstrate you the covalent catalysis using serine proteases that all have serine in the active site. So the CH2OH group side chain of the serine is essential for the catalysis of peptide cleavage by trypsin, chymotrypsin, and elastase. These three serine proteases, they have the same catalytic mechanism but differ by specificity, which you memorized before. So they cleave after different amino acids but have this all the same common mechanism. For example, trypsin cleaves hydrolyzes amide bond after lysine and arginine residues, and the reaction is shown here. So basically water hydrolyzes this bond. Serine proteases are endopeptidases, which means that they cleave internal bond in peptide chain rather than removing one by one amino acid from N or C terminal, which do hexanucleases. So you're familiar with acid catalyzed hydrolysis of amide bonds, and I have to say that this reaction requires harsh conditions. Basically, a protein should be boiled in concentrated HCl for hours in order to achieve cleavage, while serine proteases can achieve hydrolysis of peptide bond under mild conditions, basically under physiological conditions. And on the diagram of primary structure of chymotrypsin, you can identify catalytically essential histidine, aspartic acid, and serine residues. And these three residues, so-called catalytic triad, is found in all serine proteases. So here is the catalytic triad bound by hydrogen bonds found in active site of serine protease. as you can see, these residues are located far away on the primary sequence, far away from each other on the primary sequence. However, when the protein is folded in three-dimensional structures, they appear to be in close proximity and interact with each other. Serine residue is bound by hydrogen bond with histidine, and histidine is bound by hydrogen bond with aspartic acid. On this slide, you can see the same catalytic site that is bound to its substrate. This is a peptide bond to be cleaved, and this R1 and R2 are the remaining portion of the peptide to be cleaved. On the first stage, the serine attacks the carbon of the peptide bond, and this is assisted by proton abstraction by histidine residue, which removes proton from serine and increases its nucleophilicity. So you can say that this part is general base catalysis by histidine, right? And what we have here is the formation of tetrahedral intermediate. Why is it called so? It is because the carbon here now is in tetrahedral arrangement. Its hybridization state is sp3, and the lone electron pair of nitrogen is released from conjugation that it used to be in peptide bond, in the original peptide bond. So as it is as the lone electron pair of nitrogen becomes available for protonation, it gets proton from located nearby histidine residue. And the oxygen on carbon is getting negative charge and falls in so-called oxyanion hole right here. In oxyanion hole, the negative charge on oxygen is stabilized by formation of two new hydrogen bonds between NH2NH group of the side chain. So now what we see here is the peptide bond is cleaved. Let me show you. This bond, after protonation, this NH2 group becomes a good leaving group, and this bond can be cleaved. And that's what we form. This is acyl intermediate, so-called. This it shows you the these two stages, like stage 2 and 3, demonstrate you the covalent catalysis when an covalent intermediate between substrate and the enzyme is formed. This is followed by the release of the N-terminus product and entrance of water molecule in the active site. So this N-terminal product is now replaced with a water molecule, and what we have here is the next step of catalysis. So histidine acts again as a general base catalyst by removing proton from water molecule and increasing its nucleophilicity towards the ester bond, carbon of the ester bond. Here we have another tetrahedral intermediate formed. So same structure, similar structure as we had it on the stage 2 of catalysis. Again, oxygen O- is stabilized in the oxyanion hole, and the bond, the ester bond right here, the former ester bond is cleaved, and we have a C-terminal product released from the active site. So please memorize this mechanism.